Let's start our scientific session. So we start with demystifying the black box and new features and old tricks to use software efficiently. Our first speaker is James Beilsten-Edmands. He is a software scientist at Diamond Light Source, and he works on the development of dials, and it was Xia too. And he will present us about approaches to processing and selecting data in multi-crystal collection strategies. So please, the stage is yours.
Great, thank you very much. Good morning, everyone. Yes, today I'm going to talk about how to process and select data for multi-crystal data collections. Okay, so this talk is not just kind of aimed at telling the latest research to advanced researchers. That wouldn't really make much sense without an introduction of where we are today and how we got there. So first I'll talk a bit about the history of data collections, then talk about modern multi-crystal data collections, and then go through a few bits of the processing, so how we can use an ensemble approach to better process multi-crystal data. I'll talk quite a bit about different levels of non-isomorphism and how we can assess this in processing, and then a few slides on the importance of uncertainties in data statistics, and also some considerations for serial still shot crystallography. So really I'm kind of presenting the accumulated knowledge over a long period of time that's been achieved through many careful methods development and experimental studies. So did you know the very first protein structures were determined by multi-crystal methods? So back in the 60s, sufficient information for structure solution could only be obtained by combining measurements from multiple crystals. So Dorothy Hodgkin, in a review article where she was talking about the work that won Kendra and Perutz the 1962 Nobel Prize, she comments that tens of thousands of reflections from 5 or 6 crystals were measured to provide the electron density distribution in myoglobin and hemoglobin. This is a really interesting kind of a review article. I'd encourage you to read it. It's open access. Just kind of gives you an idea of how different methods were back then. I particularly like this quote. So the spectra were recorded photographically and the intensities were estimated by eye. So we've come quite a long way since then. Okay, so the first studies were multicrystal, but it was quite quickly realized that cryocooling would be necessary in order to achieve, to extend the lifetimes of the crystals. And this was demonstrated first in 1970 by Haas and Rossmann. They showed that if you collect at minus 75 degrees, this reduced the radiation damage by a factor of 10-fold. So then this gave a demonstration that you could, in theory, collect complete data from a single crystal. It then took over another 2 decades of methods development and experimental development until this cryocooling technique became standard. So by the mid-1990s, this was then the standard means of collecting data at synchrotrons, and this is due to a number of advantages that cryocooling brings. So as the synchrotrons got brighter, cryocooling reduced the radiation damage by lowering the diffusion of radicals which cause damage within the protein structure. And you get about a 70-fold reduction. In addition, the cryoprotection process and crystal mounting and handling typically reduced physical damage of the crystals compared to other methods. And there's also the potential for reducing dynamic disorder, which can get frozen out. So for certain systems, this can sometimes lead to high-resolution structures as well. However, that's not the end of the story. And like all good trilogies, we had a return. We've got the return of the multi-crystal collection. Because as you know, life doesn't occur at 100 Kelvin. So if we could collect before the onset of radiation damage, then we would ideally like to collect at room temperature to get a better handle on the biology. And this really kind of was revitalized by the advent of the X-ray free electron laser. So in 2011, Chapman demonstrated the structure solution by taking millions of still shot images of photosystem 2 membrane protein with the femtosecond pulses of the X-FEL. Now, X-FEL studies are really kind of the logical limit of multi-crystal data collections, but the zero accumulated dose that you get from that isn't a requirement to answer most biological questions. Damage typically occurs in a particular order. So first you get metal site damage, then disulfide bridges elongate and break. You get decarboxylation of glutamates and aspartates, tyrosine residues lose the hydroxyl group, etc. So as a result of this, you can collect partial rotation data sets at a synchrotron. They give you incomplete measurements within a particular dose budget. Then you can combine many data sets together to give completeness and sufficient signal to noise ratio. And then in addition to this, we've also got synchrotron still shot crystallography. And this can be complementary to X-FEL techniques for dose-sensitive or time-resolved studies. Okay, so given that we want to collect data from multiple crystals at room temperature, what's the best way to go about this? How can we make sure we have a high-quality data collection? Well, at the CSP4 study weekend in 2018, I believe, Graham Winter gave a talk and subsequently published his article on how best to use photons. Now, for a single crystal with a modern photon counting detector, the best strategy is to do a high multiplicity low dose data collection and changing between orientations if you have a multi-axis goniometer. And if you don't know the sample lifetime, you can do a step transmission approach to gradually increase the transmission, and then you can cut back once radiation damage becomes apparent, and you can do this because you have the high multiplicity. In addition to this, there were some theoretical and experimental studies that indicate that photoelectron escape happens more readily at higher instant X-ray energies, and you get better photoelectron escape in smaller crystals. So this is another means to reduce the radiation damage and extend the crystal lifetime. So what are the implications for multicrystal data? Well, if the crystals were all isomorphous, in the absence of radiation damage, the same number of scattered photons from multiple crystals would give approximately the same data quality as that was scattered from a single crystal. However, we do have radiation damage. Sorry. We do have radiation damage, so actually the average accumulated dose in a multicrystal data set is lower. This is good. Now, if we've got randomly oriented crystals, this then gives us full sampling of reciprocal space. If we measure many small microcrystals compared to a large crystal, this is likely to be further advantageous in terms of reducing radiation damage. Then still shot data collections are kind of even better in this regard because it even further reduces or eliminates radiation damage. However, we do have an issue that the data processing is kind of fundamentally different. In order to integrate the data, because we've only collected a single diffraction image that's still, we don't measure all of the reflection spots. And so because we're not at the exact diffracting condition, what we measure, it depends on the partiality of the spots. This depends on the wavelength spread, the spot shape, the orientation of the unit cell. So this gives us kind of an additional level of uncertainty. So unless we can fully model that, then the still shot data has higher uncertainty for a given number of scattered photons, and this can potentially reduce data quality and make the isomorphism assessment more challenging. So obviously the conclusion is that then obviously the best data collection strategy is the one that helps you answer your particular biological question with the resources that you have available. But there's a strong case for multicrystal approaches if you can overcome the effects of non-isomorphism. Okay, so the modern multicrystal collection is quite different from those initial ones in the 50s and 60s. One of the most common types is the in situ multicrystal collection. This is where you have some kind of physical constraints which limit the rotation range that you can achieve. So typically, if the sample is held in a grid or a plate, you have a limited rotation range where you can only accurately measure one crystal rather than getting overlaps of different crystals in the beam. You've got to take into account background effects and the effect of the actual focusing in the sphere of confusion of the experiment. This is what you typically have for VMXI experiments or I-24 or VMXM at Diamond. Now, that's not the only way to do multicrystal collections. You can do pin-mounted multicrystal collections as well. So this is where you limit the rotation range in order to reduce radiation damage. So you could do this on I-24 as well. Now, this is going to be slower than the in situ collections, but you have the potential for higher data quality from each individual sweep because you can tailor the experiment to each crystal, so you can change the beam size, you can reduce the background that way. Then still shot collections are another form of multicrystal collections. They are typically collecting on microcrystals in situ, in grids or chips, or in a viscous jet stream. So in my talk today, I'm going to mainly cover methods for processing and evaluating multicrystal rotation data sets. Now, most of these are also applicable to processing still shot data as well, where there exists the extra challenge of modeling the partiality. So how do we process data? Just as a reminder, this is kind of the standard processing approach for a single sweep. This will be the same no matter what program you use or what pipeline you use to run those programs. So first we have spot finding, where we find the strong spots on the diffraction images. We then index those with a crystal, a unit cell, lattice and orientation, as well as refining some of the experimental models like the detector geometry. Then we need to integrate the data to get our integrated intensities. We can then perform some kind of symmetry analysis to discover the symmetry. And then we scale the data to correct for experimental effects and then merge the data together. And when we then go to process multicrystal data, we have then two options. First of all, we can just treat each crystal as independent, so do independent data integration of each sweep. then do a combined data reduction with some additional analysis to address non-isomorphism. A second option that's available is to treat the process as an ensemble, so do an ensemble data integration, and then do the same kind of data reduction. Okay, so what do I mean by ensemble processing? I'm going to give an example of how you do this with dials. I'm sure you could do the same thing with XDS programs as well. But as far as I'm aware, there's no kind of automated pipelines to take this approach at the moment. This is some example data I've got, just insulin crystals, 5 degrees of rotation, so a very narrow wedge. And the advantage here is that you can basically learn, you can discover things about your data during the processing and use that to increase your indexing and integration success rate. So we define the spots as normal. We'd index each crystal with an independent orientation matrix. Then if we look at the distribution of unit cells, I'm not sure how well you can see this, but 22 out of the 24 index with the same cell parameters, 1 indexes with different parameters, and 1 doesn't index at all. So by doing this indexing, you can obviously find the most common cell, then go back and index the data specifying that unit cell. And this time we find that all 24 lattices are successfully integrated. Now this is a high symmetry example, so we only found 1 extra one, but typically, the success rate of indexing and integration is far below 100% for narrow wedge multicrystal data. So by taking this approach, we can learn things as we go and increase those success rates. We can also look at some of the other statistics you get during processing, like from the refinement step. So like looking at the RMSDs, give us some more indication of how similar the different sweeps are and whether we have some sweeps which might be lower quality if we don't index in many spots or the RMSD is much higher. Then for the data reduction, this is kind of the same whether you do the ensemble approach or integrate each one independently. You can do the symmetry. First, you have to do like this symmetry discovery process. Now, each diffraction pattern for narrow wedge sweeps doesn't have enough information to reliably assess the symmetry, but we take an ensemble approach to discover the symmetry by taking into account all data at the same time. So this is what the dials cosin program does, and it can also resolve indexing ambiguities. So it tests the different symmetry operations that are possible by the lattice and scores them and gives you a most likely point group. After we discover the symmetry, then the data can be scaled, where we correct for experimental effects. And after we scale the data, then there are a number of different approaches we can use to assess non-isomorphism. Okay, so one of the most kind of obvious forms of non-isomorphism is unit cell non-isomorphism. We can assess this with hierarchical clustering via a dendrogram. So a dendrogram is just a hierarchical structure where we join data sets into groups, and we need to define some kind of distance between the different groups. So if you wanted to do spatial clustering in 3 dimensions, you typically use like the Euclidean distance. Unit cells are a bit more complicated. You have 6 degrees of freedom. You've got the cell axes, but also the angles between them. So the way you have to do this is to define a special function. So Andrews and Bernstein define this NC dist function. Basically take, it has to be done in a 6-dimensional space and take into account the symmetry as well. Okay, so just looking at this on a few example data sets. The one on the left, this is a highly isomorphous data set. So you get some of this kind of branching structure, but there's no obvious kind of clusters within that. However, this second example on the right is from some serial data, copper nitrite reductase, where you get 2 different isoforms. And you can see here there's kind of 2 different distributions in the histogram, and there's a very large height difference between the 2 clusters. So all the data sets cluster well into 2 clusters at the low level, and then they kind of join at really high distance. So these ones kind of cleanly separate. Yeah, so that can be run with the kind of dials cluster unit cell program. I'm going to kind of put the names of different programs with what you can, how you can do this in dials on the slides as appropriate. Okay, so given that kind of radiation damage is a key driver for multicrystal experiments, we require different means to assess these. And there are 2 metrics that were developed for this, the RD, the dose-dependent R factor, and the RCP, which is the cumulative pairwise R factor that depends on dose. I think it's easy to explain these with a few examples. So here I've got some sweeps of insulin crystals. And first of all, on the left is the RD metric. So this shows you how well things agree based on a particular dose difference. But this doesn't kind of give you a clear indication of when the radiation damage actually starts. That's why the RCP metric was developed. And the interpretation of the RCP metric on the right is that as you add more data, if that data agrees less well on average with the existing data, then the RCP number increases. So on the bottom right plot, you see that it quite steeply increases for the 50% transmission data, whereas above it at 20%, the slope is much less steep. Now this is, that's kind of useful to kind of get an indication of radiation damage, but it doesn't tell us which bits of the data are actually most contributing to that. So another means to assess this is to use the delta CC half metric. The delta CC half is defined as the overall CC half taken away, and you take a subset of the data away and you take that CC half value away from the overall CC half value. So if delta CC half is negative, it means a small subset of the data negatively contributes to the overall CC half. And then removing that will increase the overall CC. So looking back at the kind of 20% transmission data from the last slide, I calculated the delta CC half on groups of 100 images. Plus as a histogram, you see a few outliers with larger negative delta CC half. 2 of those are at the end of some of the sweeps, which is likely radiation damage. And one of them is the beginning of sweep 12, where in fact the crystal was out of the beam. So that's kind of useful for kind of finding small regions of data that disagree most with the rest of the data. And you can do some iterative cycles of scaling with delta CC half to kind of slowly improve the data. So that's kind of good for looking at small regions of the data, but there's kind of a more a different method that you can use, which is the hierarchical clustering, so taking a dendrogram-based approach based on the intensities. So this was first demonstrated by Giordano in 2012, where they used this to select an isomorphous subset from a multicrystal data set. And in this case, we need the distance metric is 1 minus the pairwise correlation coefficient. That's our measure of similarity. So basically what you do is you calculate a matrix of correlation coefficients between all pairs of data sets, and then you build up the dendrogram in that way. This was first implemented in the Mesh and Collect workflow, ESRF. We've more recently added these kind of features into Dials and ZIA2. So in these examples on the right, the first one shows this kind of typical kind of decaying dendrogram structure where you've got a few data sets which disagree most with the other ones. And then on the right-hand side, this is for some distinct isomorphous subclusters where you get 2 clear regions of the data which agree well with each other. And then they disagree less well with the opposite group. Okay, so kind of that's good, but the problem there is that it doesn't help us to distinguish between systematic and random differences. So the way to do this, well, it was discovered by Bremen Diedrichs when they were trying to resolve the question of indexing ambiguity resolution for serial data. So to assess systematic and random differences, you minimize this function, where R is the correlation coefficient between pairs of data sets, and X is some coordinates that you have to optimize. For indexing ambiguity resolution, you basically make 2 copies of the data in the 2 indexing modes, and then you do this minimization process, and systematic differences get separated out in angle, and then the distribution in the radial direction is indicative of the random error. Now, this is a very powerful approach. It was used by Gilday and Winter more generally to do symmetry discovery in the COSIM procedure. And it can also be used to find isomorphous subclusters of data. So here is just an example of how we use it in Dials with the COSIM procedure. So this is some insulin data of bovine and human insulin, 8 sweeps each. And if you do the COSIM clustering, you'll see there are 2 very tight clusters on the left-hand side, which are well separated in angle. So then you can do things like dendrogram clustering based on angle to give a clear separation in the dendrograms in the first image on the right. And then this is kind of much more clear compared to the correlation coefficient clustering, where you do still have the separation into the 2 clusters, but it's much less well separated because you've mixed in the systematic and the random errors there. Okay. And kind of more recently at Diamond, we've been working on this further, and the question we're trying to answer really is how can we automatically choose some isomorphous clusters if we don't know how many clusters we're going to have, and can we do this in an automated manner? So this is something that Amy Thompson from BMXI has been working lots on, and because we have COSIM coordinates that we've optimized, well, coordinates naturally lend themselves to machine learning methods of density-based clustering. So you can use there's an algorithm called dbScan, which helps identify kind of core regions of high density and then expands clusters out from those. And optics clustering is then a development on that, which can better handle clusters of different density, and it defines this concept of reachability. So you can kind of see how closely related points in a cluster are and how much you have to then go to reach the points in the next cluster. So then this is an example from a recent paper that we've got under review at the moment. But the kind of functionality is already in dials. It's 12 sweeps each of insulin again, human, porcine, and bovine insulin. And we see that these kind of separate out very nicely in the COSIM clustering plots. And then the optics can automatically identify the different clusters there. Okay, so I just want to spend a few minutes on talking about data quality metrics as well. So something that quite kind of we've realized has become important for multi-crystal data is correctly taking into account the uncertainty. What I mean by this is the CC half formula in its standard form only looks at the absolute difference of the intensities from the average. Whereas, in reality, we have intensity uncertainties, and low resolution data has a higher absolute intensities, but it also has a higher absolute uncertainty as well. Whereas we can in fact weight the different elements of the CC half formula by the intensity uncertainty. And we find this becomes important when applying the metrics to noisy statistics, and I'll give you a few examples now. The first one is the reliability of the CC anon calculation. So this plot here is from a recent paper by Griezmann, some sulfur-SAD anomalous data on lysozyme. They originally processed it with dials and they made the raw data available, which is very handy, so I could reprocess it. But if you look at this figure from the paper, you see that the CC anon is kind of quite high at high resolution, and it kind of weirdly dips down at low resolution, which doesn't really make sense. So kind of reprocessing the data with the latest dials, first looking at the kind of delta F over F plot, which is an indicator of anomalous signal, that kind of looks as you'd expect. There's no kind of deep dip at those resolutions. Calculating the CC anon with the standard unweighted formula that we usually use, we again see this kind of big dip at low resolutions. Now processing exactly the same data, but we're using a weighted CC half formula. We then get this plot on the right. So actually that gets rid of this kind of unusual dip, and it kind of makes much more sense and is now consistent with what we'd expect from delta F over F. So the kind of true correlation was kind of being hidden by using unweighted statistics. And if we just kind of quickly look at why that's the case, if we plot the data on the left which is delta, this is the data that goes into CC half formula, delta I over I, you see there's kind of quite a wide distribution. Some of those are kind of in the wrong quadrants from what you'd expect based on positive correlation. But once you add in the uncertainties, you see that actually they have a high relative uncertainty. And if you were to look at the parts of the data set with the highest relative weights, when you do a weighted formula, you see that actually they lie much more along the kind of positive correlation line. So the kind of high uncertainty of some points is kind of obscuring the overall correlation in the data. Uncertainty is something we use as routine when we're taking the kind of data downstream into structure determination. So I think we should be taking this into account when we're calculating these statistics. It really kind of only makes a difference for multicrystal and serial data where there's higher levels of noise. As a second example, if you're looking at these kind of correlation coefficients as part of like a COSIM or Brem-Diedrich procedure, that makes a big difference as well, because you've only got a few reflections that overlap and they have a high relative uncertainty. So here's another example from our kind of recent work that's under review. This is 216 crystals, 60-degree rotation, and we find that we kind of only get useful clustering either from kind of dendrogram-based approaches or the kind of COSIM optics clustering if we use uncertainty weights. Otherwise, we just find all the data is mixed and we don't see any kind of structure within the CC matrix. So, uncertainty weighting is important for getting kind of accurate CC half metrics, and kind of a similar kind of weighting was applied to the sigma tau version of this formula in this paper based for XDS CC half. Okay, so last few minutes. So I just want to say that kind of I've shown a few programs that we can use in dials. These are all kind of grouped together in the multiplex pipeline. So there's the option to do kind of hierarchical clustering based on correlation, cosine angle, or the delta CC half scaling and filtering. And we've recently added a few new options. So you can now do this optics-based automated clustering in multiplex. We've reworked some of the program options to be more intuitive, and we've separated the clustering aspects into kind of a standalone program, so you don't have to rerun the whole pipeline. And then the considerations for still shot crystallography are that lots of these things apply. So you really kind of need that Brem-Diedrich approach to resolve indexing ambiguity. And as the uncertainty increases, then that's really important to take into account when you're using these different metrics to make decisions on which subsets of the data to keep. And I think this kind of isomorphous intensity clustering is kind of the most promising approach for selecting subsets of data from large serial data sets. something I plan to investigate more. And just in terms of kind of the fundamental difference for integrating still shot data, we've been doing some work on this in dials as well. We've done some more advanced modeling to try and better understand what the partiality is of spots. So we've got some recent work on this, and there's also similar approaches being added recently to CRISFEL and some other modeling by Mendes. So kind of different groups are trying to address this problem of the higher uncertainty in still shot data. So perhaps that will become less of an issue. So in terms of what are the kind of new tips and old tricks, going back to the theme of this session, well, the ensemble processing is a way to increase your success rate in data integration. It doesn't need to be much more complicated than processing a single sweep. There's different ways you can assess non-isomorphism. There's been quite a lot of algorithm development on this in the past 10 years, and now there's a number of different tools available to help you select your isomorphous subset. We have these kind of utility programs within dials, but there's other programs available as well, and other pipelines being run at facilities to help you make these decisions. So we've got this kind of now powerful set of multi-crystal techniques to answer particular biological questions, but it does require some knowledge of the data processing to perform the best experimental analysis. Okay, so I just wanted to kind of acknowledge that lots of people have contributed to different aspects of this work, and obviously I'm presenting work done by many people around the world. I hope I've made that kind of clear by my references. Particular thanks to kind of the dials team at Diamond and the MX software team who have kind of helped to make these ideas into useful products. And thanks for the collaborations we have with the Beamline teams as well for giving us nice data and extensive testing and feedback on our programs. Thank you for your attention.
Thank you very much, James. Do we have any questions?
Thank you, James. That was a terrific talk. Just a couple of remarks. The first one is just a typographical point. I think in many places you've written CC1/2, where you just meant to write CC, the formula for the Pearson correlation coefficient. The 1/2 comes a bit, has a specialized meaning. Another thing about this uncertainty weighted correlation coefficient, Ian Tickle was adamant that this is the way correlation coefficients should be computed. So this has been implemented on the StarNISO server for many years. And it's used in producing one of the three-dimensional plots on CC1/2.
Okay.
Yep. But terrific developments. Thank you.
Yeah, thank you.
Hello. Thanks a lot for your talk. And this is a naive question, but the COSIM distance for clustering and optics and DB-scan methods of also grouping datasets, are they already available in the latest distributed version of dials?
Yes, they are. Yes, the difference is the kind of the COSIM procedure kind of to optimize the kind of Bremner-Diedrich formula to get the coordinates. Then we do the density-based clustering on those coordinates. So they kind of, that's like an extra add-on, I guess, to that kind of procedure. Yes, we recently made that available in dials. So if you got the latest version, you can try the dials correlation matrix program to look at those spots.
Any other questions? I have actually a question about your ensemble processing. Is this like the default, or do you have to specifically set that up?
No,
do you recommend trying both and seeing what works better?
So the ensemble processing at the moment is something you'd have to do. You'd have to script yourself with the dials programs or XDS programs. The automated processing pipelines typically treat each sweep independently. So you'd have to set that to yourself.
Are there any questions on Slack? Could anybody? Otherwise, if there are any questions on Slack, please check it and try to answer those as well.
Okay.
So then otherwise, let's move on and let's thank James one more time.
Thank you.